What's up, guys? So our project is called Breathe In, Cough Out, and it is me, Madison, Trevor, and Hannah. And our original project was going to be taking plates and putting them inside sorority and fraternity houses and placing them outside also those houses to compare the air quality inside versus the air quality outside and see what microbes were growing. But since we can't do that, we have to evaluate other studies that did something similar in counting bacteria in the air. But before we get to that, why did we choose this project? Well, air is an important thing in society, and since we breathe it every day, we want it to be clean. Oftentimes, the air is not as clean as we think it should be, and oftentimes we do get illnesses from this air. In this project, we wanted to see how safe our air really is and if air filters really do work in purifying the air. The inspiration for this was the sight of mold in some of our buildings that we live in respectively, like a fraternity house and a sorority house. We have seen mold. We wanted to see if there were other microbes around the air filter that we don't know about. And lastly, we wanted to also see how air filters work and see if they actually do work. A lot of illnesses nowadays do come from the air, especially given the situation that we're in with COVID-19, which has been proven to spread through air droplets. We want to make sure that our air is purified and clean so that we're able to use it efficiently and not get sick from something that should be so simple to control. In this project, we investigated a few articles that talked about air quality and did a bacterial count of the microbes in the air, and we're going to use that to kind of predict what our project would have been like. So next, we're going to have the hypothesis, and that will be Trevor. He'll be doing the hypothesis and the methods.
Hey guys, the purpose of our study was to assess the air quality in different buildings, and in our purposes it would be a fraternity house and a sorority house, and see if there was any correlation between the higher air pollutants within these environments and certain respiratory illnesses. Our study is actually very similar to one done by Zudu Anamalman. I don't think I got that last name right. But essentially his study looked at the air quality within different primary schools, which I'm sure are just elementary schools in different countries. And the purpose of his study to test the air quality of these different classrooms is based around the fact that due to higher occupant density, poor sanitation of classrooms, and insufficient air supply would cause respiratory illnesses. Our study also was to assess the air quality within fraternity and sorority houses and see if there was any correlation between the air supply of these buildings and certain respiratory illnesses. Now, another study done by I'm not even going to try his last name. But his study actually assessed the air quality within ambulances. He tested the air quality within ambulances before they went to receive a patient and during a patient service within an ambulance. And his study actually also was looking to see if there's any correlation between symptoms of influenza, SARS, and pulmonary tuberculosis, as well as others, to see if there's any correlation between the poor air quality and these respiratory illnesses. Now, just to be clear, we couldn't really have a way of knowing if the air quality directly correlated to causing respiratory illnesses, such as the ones that I just described. There is definitely a possibility that the air quality causes these illnesses, but we have no way of knowing that for sure, for our study at least. For their studies, they had way more resources than we would have had. We would have just predicted that the air quality within fraternity and sorority houses may cause respiratory illnesses in the future for those who are in these buildings a lot or whatnot. The methods between our study and these two other studies are similar in some ways, but vary in data collection ways, which I'll get to in a little bit. So for our study, we were going to place 2 TSA plates inside and 2 TSA plates outside of a fraternity and a sorority house. Now, our TSA plates would collect microbial fallout from the air within and outside the fraternity and sorority houses, and this would be our quantitative data that we would then use enumeration to see how much air pollutants are present on these TSA plates. We would also have 2 PDA plates placed close to mold growth within each fraternity and sorority house. And yes, both of them had mold growth in them, and it was disgusting. So we thought that having that PDA plate there to catch the microbial fallout from the mold would actually add to our study and incorporate qualitative data as well as quantitative data to reaffirm our hypothesis of pollutants in the air being present. For the research study done in different classrooms amongst various primary schools, this researcher exposed petri dishes filled with blood agar in 51 different classrooms amongst 8 different primary schools. This procedure is very similar to what our procedure would have been by exposing the TSA plates inside and outside fraternity houses, and the essence of exposing these dishes was to collect the microbial fallout that would have been present within these environments. Now, a rather expensive and rather complicated method done by the researcher who assessed the air quality within ambulances used a biostage impactor quick take 30 sample pump. Now, this mouthful is rather expensive and I'm sure caught way more airborne microbial particles in ambulances than just exposing a petri dish, but nonetheless, either tactic at the end of the day caught any microbial airborne pathogens that were present within. The data collection for our study and these 2 studies are a little different. For our study, our data collection method was going to be enumeration by taking samples from the TSA plates and running them on a petri film to see how many microbial life forms were present within the TSA plate. That would be our quantitative data. And our qualitative data would have just been the results from the PDA plate and that mold growth shown hopefully on that plate, just to reaffirm that there are various airborne pathogens present within each environment in the sorority house and the fraternity house. Now, our data collection is different from these 2 studies in a sense that each of the studies involving the primary schools and the ambulances actually ended up doing bacterial identification processes such as gram staining and colonial morphology to assess what particular pathogens were in the air. Our study wasn't going to go that deep. We were just going to see how many pathogens were present in a sample environment, whereas these studies were seeing how many and what kind of pathogens were present within each of these environments. So that is a little difference in the data collection between our study and their study, but nonetheless, at the end of the day, both our study and the 2 studies that I just previously talked about both looked at microbial fallout in different environments and seeing if there's any correlation between this and any respiratory diseases that could occur from these airborne pathogens.
Okay, so for the data, the first study was Andy study, and he was reporting the bacterial counts inside different public schools. He went to 8 public schools, and he recorded the bacterial count in the morning and in the afternoon, and then totaled it up. And between the 8 schools, the total in the morning was 2,826 CFU per cubic meter, and then versus in the afternoon, the total was 4,514 CFU per cubic meter. So this is interesting because we see an increase here. And what's even more interesting, and this is something that we would have done too in our project, is he recorded some physical properties that were occurring at the time, including the temperature and the humidity. So during the time that he was recording this, the temperature increased from 12 degrees to 24 degrees Celsius, and the humidity, and that was obviously increasing in the afternoon, and the humidity ranged from 14 to 64%. And these are both factors that we definitely would have seen when measuring bacterial counts at the University of Florida, as the state of Florida has very varying degrees of temperature and humidity that we see throughout the year. So we definitely would have kept track record of that as well. And then moving on to data from the second experiment, we have the study that reported the difference in isolated colonies, as well as bacterial counts between the inside and the outside of an ambulance, both before and while it was running. However, the only clear data that they have on inside versus outside is really just on isolated species, but that still gives us some insight into what our results would have been. So here they found that the majority of what they were able to isolate was staph A, which also coincides with what the first experiment was able to isolate. They both isolated a majority of staph A, so we probably would have seen something similar. And then, however, as we're able to see here in the table 3 that's provided on our poster project, it seems that the filters were able to, they detected more fungal species outside versus inside. However, the bacterial species, there seems to be more inside the ambulance versus outside the ambulance. But this case might be different as it was an ambulance and they're bringing in people who were bringing in bacteria. However, our houses still would have had people walking in and out, so we might have seen similar results.
The results of the bacteria in primary school study showed that the bacterial load increased from morning to afternoon. It was noted that the bacterial counts increased as the humidity and temperature increased. In the ambulance study, the mean and standard deviation of bacterial and fungal counts at the start of the ambulance runs were 468 and 656 CFU per meters cubed respectively. It was found that the distribution of genus or groups of isolated bacteria and fungi from in-ambulance air was proportional to the air from the outside. The study states that due to the high levels of microbial counts found in some ambulance runs, the air ventilation in ambulances should be improved. These studies reveal that humidity and temperature correlate with bacterial counts, and that high bacterial counts can be a result from poor air ventilation. Based on these results, we predicted that our research would have shown that the bacterial counts inside the houses were lower than outside of the houses, assuming that the air ventilation was properly maintained, due to the increase in humidity and temperature outside.
All right, and that is our project. So in conclusion, what we found in this project was that most likely would have been seen in ours is that we would have had a smaller and less bacterial count inside the houses versus outside the houses. That can be due to a few things like humidity and temperature inside versus outside the houses, as well as the air filters doing their job. However, we can't be 100% certain of what our project would have been like since we never actually did it. But this study kind of gives us an idea of what we would have been looking at in terms of bacterial count and other things. Hope y'all enjoyed it. Stay safe out there.